All right, let's just jump right into this explainer. Today, we're tracing the absolutely incredible iterative evolution of CRISPR. I mean, we're talking about how scientists took this primitive bacterial defense system and systematically engineered it into a highly precise, multifunctional molecular toolkit. If you've ever wondered how we leveled up from essentially just randomly breaking DNA to using a literal biological word processor, you are definitely in the right place. This whole breakdown is based on a massive milestone 2024 paper in Nature Reviews Molecular Cell Biology by Lucas Villiger and his team. Think of this explainer as your master manual. We're going to follow the exact chronological story, from the initial problem to the biological limitation and finally the innovation that took precision genetic medicine from a total sci-fi dream to an actual clinical reality. Because, look, here's the fundamental issue. Reading the human genome, fast, cheap, and honestly pretty easy nowadays. But editing that actual blueprint of life, that was a massive wall for decades. Making even one single wrong cut among 3 billion letters of code could be absolutely catastrophic for a cell. So fixing just one mutated letter to cure a devastating disease meant we had to engineer our way out of one biological limitation after another.
So here's our roadmap for the journey today. First, the blunt scissors, then pencils and word processors, heavy machinery for large genes, epigenetic dimmer switches, editing temporary RNA messengers, and finally, the CRISPR evolution and final test. Let's get right into it.
Part 1, the blunt scissors. This is the first generation of CRISPR. We gotta start our timeline with the standard Cas9 enzyme, borrowed straight from a bacterial immune system. Think of this complex like a really aggressive brute force pair of scissors. Cas9 scans the genome looking for a specific anchor sequence, what we call a PAM. Once it locks on, it physically unzips the DNA to form a little bubble and uses two specific cutting domains, HNH and RUVC, to just completely sever both strands. It creates a blunt double-strand break. At a molecular level, it's incredibly good at finding its target, but it's fundamentally destructive.
And that destruction is the problem. By relying on double-strand breaks, we're essentially forcing the human cell into full-blown panic mode. The cell urgently tries to glue that DNA back together using this error-prone repair pathway called NHEJ, and that leaves messy insertions or deletions, literally these little scars that just knock out the target gene entirely. Now, if we actually want a precise cure, we try to hijack a different pathway, HDR, to use a repair template. But the reality? That process is incredibly inefficient in most adult, non-dividing human cells, so relying on double-strand breaks often gave us toxic, totally unpredictable mutations instead of the clean cures we needed. Which brings us to Section 2, pencils and word processors. We had to eliminate that double-strand break entirely because those breaks were causing such toxic scars. Bioengineers had to evolve the tool. Enter base editing. Instead of fully cutting the DNA, they mutated Cas9 into a nickase, basically dulling one of the blades so it only snips a single strand. Then, they attached a chemical enzyme called a deaminase to it. This acts like a chemical pencil. We can now perform direct chemical reactions on individual letters inside this tiny, maybe 4 to 8 base pair editing window. We can literally just chemically erase a cytosine and turn it into a thymine, or an adenine into a guanine, without ever shattering that delicate DNA backbone. It was an absolutely stunning leap in precision. But base editors definitely have a catch. They're basically chemical pencils that are limited strictly to specific transition letter swaps. And here is the fatal flaw. If you have multiple identical letters sitting together inside that tiny editing window, you risk making unwanted bystander edits. You end up altering DNA you definitely meant to leave alone. Plus, what if you need to rewrite a whole sentence instead of just erasing one single letter? Well, that exact limitation is why scientists pushed forward and invented prime editors. Welcome to prime editing, our molecular word processor. We take that same Cas9 nickase, but this time we pair it with a reverse transcriptase engine and a highly specialized guide RNA, a pegRNA. This guide doesn't just act like a GPS to find the spot, it actually carries the exact replacement text right on it. The reverse transcriptase enzyme literally reads this template and writes brand new DNA code directly into the genome, which displaces the old mutated strand. It is quite literally a search and replace function for genetics. It unlocks all 12 possible base swaps and small insertions with just astonishing accuracy.
Moving to Part 3, heavy machinery for large genes. It's time to install whole software patches. So prime editing is fantastic for those tiny, small fixes. But what happens when you hit a size limit? Let's say a patient needs an entire gene inserted, not just a few scattered letters. Prime editing efficiency just completely plummets if you try to insert anything larger than about 50 base pairs. For diseases caused by hundreds of different mutations across a single gene, scientists realized they couldn't just sit there and fix one typo at a time. They needed heavy machinery to drop in massive, completely functional gene payloads. They needed an entire software patch.
And that brings us to the PASTE system. This is so cool. The prime editor goes in first as basically an advanced team, and it writes a tiny little landing pad into the genome using its search and replace mechanism. Then, a viral enzyme, an integrase, locks on to that exact pad and just drops in an entire gene payload. We're talking up to 36,000 base pairs long. By splitting the job into two parts, we get the incredible precision of prime editing combined with the brute payload capacity of an integrase. We bypass that 50 base pair wall completely, and we do it without a single double-strand cut. Section 4. Epigenetic dimmer switches, changing the game by regulating gene volume. Now let's step into a totally different dimension here. What if we could change a gene's activity without altering the DNA letters at all? The huge breakthrough here was realizing, hey, we can use a completely dead Cas9. Since it can't cut DNA anymore, we just use it as a super targeted delivery vehicle to carry volume controls directly to specific genes. We're tapping into the epigenome, depositing chemical methyl tags onto the DNA or its surrounding proteins. These tags turn a gene's expression up or down, literally just like a software dimmer switch, and they leave the underlying genetic code completely untouched. And we've actually made a massive leap here from temporary to permanent control. With early tools like CRISPR, we could temporarily block the gene, but it would eventually turn back on. But with CRISPRoff, we deposit permanent methyl tags. This means even after the CRISPR tool itself degrades and completely leaves the cell, the target gene remembers that it's supposed to be off. It passes that epigenetic memory down through over 450 cell divisions. We can now silence disease genes permanently, herdably, and safely, without ever touching a pair of molecular scissors. Part 5. Editing temporary RNA messengers. Let's look at transcriptome engineering. But sometimes even epigenetic memory is way too permanent. What if we want a treatment that does its job and then completely vanishes? Well, instead of editing the master hard drive, the DNA, we can target the temporary printouts, the RNA transcriptome. Because messenger RNA naturally degrades over time in the cell, editing it acts a lot like a temporary medication. It gives us this incredibly safe, fully reversible way to treat acute cellular issues with absolutely zero permanent genomic risk. But of course, there was a hurdle. Early RNA editors had a really massive flaw. The enzyme they used, Cas9, acted kind of like a grenade. Once it found its target RNA and cut it, it just kept going, indiscriminately chopping up nearby healthy RNA, which caused severe cellular toxicity. So to fix this dangerous collateral damage, scientists went back to the drawing board and engineered a vastly safer tool, Cas9. Instead of a grenade, Cas9 acts like a highly trained sniper. It cleanly degrades only the specific target messenger RNA with absolutely zero collateral damage to the rest of the cell. And finally, Section 6, CRISPR evolution and final test. Let's pull this all together. We've climbed the complete evolutionary ladder of CRISPR now. Looking at this whole tech tree, you can really see the continuous story of problem solving here. Standard Cas9 caused toxic double-strand breaks, right? So we made base editors to stop the cutting. But then base editors couldn't swap every letter and caused bystander mutations, so we built prime editors. Then prime editors couldn't insert whole genes, so we engineered PASTE. Literally every single molecular tool you see on this breakdown was explicitly invented to solve the fatal biological flaw of the one right before it. So let's test if we truly understand these mechanisms with a quick Feynman technique challenge. Think about it for just a second. How exactly did scientists solve the specific cargo size limitation of prime editing? Well, remember that 50 base pair wall? By combining a precision writer with heavy molecular machinery, scientists bypassed it entirely. Prime editing was basically relegated to just writing a tiny landing pad. Then the serine integrase steps in and does all the heavy lifting, pulling massive gene payloads up to 36 kilobases smoothly into the genome, entirely without breaking the DNA. We are no longer just passive readers of the biological blueprint. We're its active authors. With these incredible tools moving into human clinical trials right now as we speak, the era of true genomic medicine is officially here. It really just leaves us to wonder, with a toolkit this precise, just how far can we push the boundaries of human health?